Right now, inside our body, nearly 37 trillion tiny living units are working together silently. They build our muscles, repair our wounds, store our memories, and even decide whether we are living or dying. But the shocking part is, every one of them started from just one cell, right? Now, I'm going to tell you something very interesting. This world is inhabited by innumerable number of species of living organisms. Can you guess the number? The number is 1.7 to 1.8 million. It's a huge number, and scientists are adding new species. They're discovering new species every single year. You can say every single day new species are found, they are included. Now one thing is common. See, all of them are made up of cell. Some of them are unicellular, while some of them are multicellular. You can also say, sir, some of them are few cell. Yes, obviously, unicell, few cell, multicellular, obviously they are. However, any living body is formed of either one or more cell, right? Now, here is my first question. What is a cell, right? What is a cell? You're going to tell me a cell is the fundamental structural and functional unit of all living beings. That is the definition we have learned in our childhood, right? Fundamental structural and functional unit. So fundamental, structural and functional unit, right? Yes, this is true. But do we actually understand what we learned? What is the meaning of structural unit? Why it is also called as the functional unit? We must know. We must know in details, right? Yes. So, welcome to everyone. This is Bioschool India, and we are going to discuss about the introduction to cell. This is the first part, and subsequent parts obviously you must watch, where we will see and we will discuss about the different cell organelles, right? Their functions, their structure, cell division and all. Now, moving on to answer this question, as I say that cell is the structural and functional unit of life. What does it mean? To find this answer, we are here. See here, on the left side, you can see a brick wall. Also, you can see a cell at the center. Now, what is the similarity here? Here we have mentioned, see, structural unit. Here it is mentioned as functional unit. Why? How it is relevant to a brick wall? A wall is made up of bricks. You can say bricks are joined together to make the wall, right? Similarly, all the living things, the few cell organism and the multicellular organisms, they are made up of cell. The cells are joined together to make their body. Whether it's a few cell organism, whether it's a multicell organism, they are made up of cell. In case of multicellular organism, there are different types of cell. But all of them, the common thing is all of them are cell. They may look different, their function is definitely different, but they are all cell. That's why we can say that it's a structural unit of life. It provides shape, support and organization. Well, good enough. Why functional unit? Because all the basic functions which keeps us alive, which make us qualify as a living being, it happens actually inside the cell. It starts at the cellular level. What are they? See, metabolism, energy production, synthesis of DNA, RNA and other molecules, transport of substances, growth and repair. All these things are happening inside our body continuously, and that's why we are alive. But all these things, all this metabolism, energy production, synthesis, transport, everything happens at the cellular level. That's why rightly we can say that cell is the structural and functional unit of life. This is very, very important. We must understand it very well. Why we are calling it as structural and functional unit of life. Okay? All right. Now, next is the cell theory. When you understand that cell is the structural and functional unit of life, then the next topic comes the cell theory. Who has given? Two German scientists. Can anyone tell me the names? Yes. One of them is Matthias Leiden. He was a German botanist. Another was Theodor Schwann, a German zoologist. What they have said? They said all living organisms are made up of cell. They also mentioned cell is the basic structural and functional unit of life. And another scientist named Rudolf Virchow in the year 1858, right? He has said all cells arise from the pre-existing cell. So these are the basic theorem, the first three postulates of cell theory that you can see. But after that, the modern version of cell theory has also come, but this is the basic. Here it all started. All living things are made up of cell, because the German botanist Sladen, he has observed many plant species and gave the statement that all the plants are made up of cell. The zoologist, he said that all the animals are made up of cell. So obviously, if all the plants and animals at that time, they did not have any idea about the Monera, Protista, fungi and all. They only knew about the plants and the animals. So if all the plants are made up of cell, if all animals are made up of cell, then rightfully we can say all living organisms are made up of cell. Cell is the structural and functional unit of life that we already know. And Rudolf Virchow added that all cells arise from the pre-existing cell. So he is the first one who has given the idea of cell division here, right? The idea of cell division. First we get from here. Okay, moving on to the next. The discovery of cell. Who are the pioneers? You already know, right? The Robert Hooke, Antony van Leeuwenhoek, Rudolf Virchow, I already mentioned. But before that, some other scientists were also there who has worked. We know detailed about Antony van Leeuwenhoek, we know about Robert Hooke, especially about their work. But there were scientists like Zacharias Janssen. He has also worked. Galileo Galilei. They have also worked on it, right? Previously they have also worked on it. Their work was also significant, but mostly we know about the Robert Hooke's work and Antony van Leeuwenhoek's work. Okay, see here we can see the Leeuwenhoek's microscope. Also in the image, if you look carefully, you can see what are the things he has observed. He has observed the bacteria, the protozoa, the algae, sperm cells. He has actually started with a drop of water, right? He has actually started with a drop of water. And the instrument that he has used, right, this one doesn't look like a microscope, but the magnification was significantly good. Although it was a single cell instrument, but the magnification was significantly good. Around 200 to 250 times magnification he produced. And the things they had observed, I just told you, bacteria, protozoa, algae, sperm cells, that he has observed. Now, what is his actual impact? Why he is still known? Why? See, first point, this one. He actually opened the door to the microbial world. He said there is a unknown microscopic world that we cannot see. They are living, but they are so small that we cannot see them. So he actually laid the foundation of microbiology, where nowadays all these bacteria, protozoa, algae, they are studied in details, and they have significant impact on human health also, right? That's why it says that it changed the way we see life. That's why that is the importance. He is also mentioned, see, father of microbiology. It's very, very important. You already know him, Robert Hooke. His microscope. First, he has used 2 lenses. You can see it here. You can see, right? 2 lenses were there, eyepiece and objective lens. What he actually observed? He was observing the cork cell of a plant. Now, present day, we know that it is, dead plant cells. But at that time, they did not have any idea on it. So they observed, draw pictures, and he called them cell. He is the first person to use the word cell. And he has written a book, Micrographia, right? From this Micrographia, the study of cell originated. You can also say from this book, one chapter, it actually gave birth to the subject, the branch of biology that we call as cytology. It actually came from the book Micrographia. He is the first person to use the word cell because he saw some empty box-like structures and he compared them with the empty rooms in the monasteries where the monks used to live. Right? So obviously it is important. He used a simple microscope, but unlike Leeuwenhoek, he used 2 lenses. He observed cork cell, then saw tiny compartments, named them as the cell. Next. As Robert Hooke has already discovered, but that microscope was very primitive, that was limited on magnification. But nowadays we have seen microscope in our school, the compound microscope we have seen in our school. Various magnification power is also present. If you look carefully, you can see the various part of the microscope, right? Various parts. The eyepiece, the body tube, revolving nose piece, objective lenses, then the stage clip, this is the stage clips, slides, diaphragm, light sources, coarse and fine adjustment, right? Coarse adjustment and the fine adjustment. So these are very, very important. We have seen this microscope in our school. Now this microscope is very, very important when we talk about the cell study. Why? Because the limitation of the human eye, 0.1 millimeter. We cannot see beyond this. So we need an instrument. Leeuwenhoek show us the way, Robert Hooke shown the path. Then this instrument is made. Higher magnification is possible. We can work due to this, light. Due to this light, we can work at night also. Now the modern microscope has a facility of taking and recording videos also. So it is very convenient to use this. You got a very good slide, you can take a picture, you can record video also. Right? In the school, colleges, these microscopes are, used variously. We have seen this, we have used this. But we have to know one thing. What is the meaning of the word micro? How small it is? We know up to millimeter. Millimeter is everybody's, everybody knows millimeter, how much is. You have a geometry box scale, you already have millimeter marks into that. But after that, smaller than millimeter is micrometer, right? We'll understand that. See, the relation between millimeter and micrometer, you have to understand this, because microscopy is integral part of cytology. Without a microscope, you cannot study. You don't understand even. So, if you're using an instrument that has the word micro, so you should understand what does it signify. So here it is written, see, the first line is explaining all. 1 millimeter, in the geometry box scale, you have the 1 millimeter mark. Identify it, take it in your hand and identify. And after that, divide that 1 millimeter into 1000 parts. Then one part become the micrometer. So obviously you can see 1 millimeter divided into 1000 parts, that gives you 1 micrometer. And human eye, how much you can say? We can observe only 0.1 millimeter. Beyond this, human eye cannot detect anything. So obviously we need a scope which can see at the micro level, and that's why we have a microscope. Next, we have seen a compound microscope, but I'm going to show you some other microscopes that are being used. See the names. We have light microscope, then we have the phase contrast microscope, dark field microscope, fluorescent microscope, confocal microscope, stereo microscope, digital microscope. You don't have to do a research on this, just know the names, see what they're used for. Here, the phase contrast microscope is observing live unstained cell. Staining we know, right? Staining is very important. We have heard about gram staining and all, G plus, G minus, yes. Then we have fluorescence, then confocal microscope, 3D images it will make. Then we have the stereo microscope and digital microscope as well, right? But their magnification is limited. They're used in various purposes, but the magnification is limited. So for that came forward two German scientists. The name is Knoll and Ruska. What they have done? In the year 1932, they have discovered this brilliant microscopes called electron microscope. In the year 1932, they have discovered this electron microscope, right? 1932. Scientist name Knoll and Ruska. In the year 1940, they are put to use. Magnification 1 lakh to 5 lakh times. Can you imagine? 1 lakh to 5 lakh times magnification they are giving you. Significantly higher than the previous microscope we have seen. So now, instead of looking at the cell, they started looking at the inside the organelles, their structures, the atoms even visible. So now it is divided into two parts. One is called SEM, one is called TEM, that is scanning electron microscope and transmission electron microscope. And what they are best for, you can see here, they are best for various purposes, various uses are there. But the thing is, the first thing they uses electrons. Electrons instead of light, they are using electrons, and magnification is 1 lakh to 5 lakh times, which is significantly higher than the previous one. Now this has opened the door for the detailed study of cell. Not only cell, I must not emphasize on cell only, but now they are being used in material science and other science also, even geology, crystallography, everywhere electron microscopes are being used. One of the greatest discoveries of this in the previous century. Now, the next one, that is types of organisms based on number of cells. I think I have already mentioned you in the at the beginning. It can be single cell organism. For example, this is, we are familiar from our childhood, amoeba, right? Then we have paramecium, euglena, bacteria. These are single cell organism, and since they are single cell organism, you cannot expect any specialized function on them. Then we have few cell organisms, example volvox, which is alga, then anabaena, yeast. We are familiar, right? This name, yeast, very familiar. Remember anaerobic respiration and the end products. Yeast is one of the most important here. Then comes the multicellular organisms. Plants, animals, fungus. They are multicellular organisms. Specialized function, cell to tissue, tissue to organ, organ to organ system, and ultimately the organism. So much more complexity, much more fine work. Then we have an interesting fact. The smallest, the longest, the largest cell. What is the smallest cell? You tell me, sir, mycoplasma. Yes, obviously mycoplasma is the smallest one, right? Then the longest, the neuron, and largest, the ostrich egg. But here is the catch. Many of the students do not know which part of the ostrich egg is actually the cell. Which part? Look at the picture. I have marked it for you. Very interesting, right? We have the smallest, we have the longest, we have the largest. But sir, you have said that we cannot see the cell. Yes, you cannot see the normal cell, which are less than 0.1 millimeter size. But some of them are exceptions, like a neuron cell. It can be up to 1 meter, right? So obviously you can see. Here it is 15 centimeter, up to 15 centimeter they have mentioned. Obviously we can see. See here, 15 centimeter, up to 1 meter, we can see. But this is not possible. We cannot see it. So most of the cell we cannot see. One single cell I am talking about. One single cell we cannot see. But there are cells which are significantly bigger that we can see. Now, I am going to give you some interesting informations here. See, look carefully. I just told you about the different types of cell, right? Smallest cell, yes, it is mentioned. Largest, longest, mentioned. Now, tell me, largest plant cell. Nobody knows? Plant cell which can be visible in naked eye. Remember this. Acetabularia. This is important. Then largest plant, eucalyptus, you already know. Longest plant cell, so these are some interesting information you should remember. Please write it in your pen and paper, using a pen and paper. Right. Moving on to the next. Smallness of cell is a greater efficiency. So in the previous slide, we just discovered, or we have seen the different types, right? Smallest, largest, longest, and some other. Now, here it is mentioned smallness of the cell is actually related to greater efficiency. We try to understand this mathematically. What actually happens? If you look here first you have to look here. See, you can see a cube which has 2 millimeter sides. Now look at this calculation. Look at this calculation carefully. Side of the cube is 2 millimeter, so surface area becomes 4 millimeter square. Any doubts on this? How much it will be? It will be 2 into 2, right? Obviously. But total faces, how many faces this cube have? Total 6 faces, right? Which means total surface area, how much it will be? 24 millimeter square. I hope you understand this one. Very easy calculation. Now the next part, here what we are doing, we are cutting that one large cube into 8 small pieces. So if we cut the cube exactly at the center, earlier it was 2 millimeter, now it will become 1 millimeter, obviously 1 millimeter. Now total surface area of one piece, how much it will be? 1 into 1, that is 1 millimeter square. Now 6 faces, each small cube has 6 faces, so 6 into 1. But how many cubes we have? Earlier we had only one large cube, now we have 8 cubes. See, which increased the total surface area into 44 millimeter square. One large cube, 24 millimeter square. When you cut it into 8 pieces, it becomes 48 millimeter square. So more surface area is actually related to greater efficiency for better exchange of nutrients, gases and waste. This is very, very significant. Understanding this concept is very, very important. Next, cell shape and function. In multicellular organism, we get to see different types of cell. Their shape is different. Why? Because multicellular organism, their function is specialized. If their function is specialized, so obviously the shape is also different. If you look carefully, we have couple of animal cell and we have couple of plant cell here. Their shape is different. They can be flattened, cuboidal, columnar, branched, even spherical, rectangular, elongated. We have also have some examples, their diagrams, their function and how the shape helps them. This is very, very important part. The cell has different shape based on their function. Please remember this. The cell have different shape because of their function. Obviously they got the shape for the best functioning. The work they are doing, the shape helps them. That's why they got the shape. That is the main idea here. Next, types of cell. You already know prokaryotic, mesokaryotic, which is intermediate actually, and eukaryotic. You already know this. What is prokaryotic cell? There is no true nucleus. Yes, genetic material is present. We call it as nucleoid, right? We call it as nucleoid, or you also call it as naked DNA, which is swimming into the cytoplasm. In eukaryotic cell, you can see a proper nuclear region, nuclear pore, nuclear membrane, genetic material, then nucleolus, nucleoplasm. All together they have a organized setup. That's why this is eukaryotic cell. And intermediate between these two is a mesokaryotic cell. The example could be a cyanobacteria. Here the example could be a bacteria, and here example you already know. Apart from these three, there are other two types of cell, all the body cell and the reproductive cell. Why they are different? Because when we'll study about the cell division, we'll see here we get to see the mitosis, and here we get to see the meiosis type of cell division. Okay. So this is the end of this discussion. I hope you liked it. We're going to bring some more videos. It is just the first part, the introduction to the cell. Then we are going to discuss about the part of the cell, different parts in details we are going to discuss. So everyone must visit this channel, and we are going to bring some interesting videos which are going to be very, very useful for your NEET preparation. Thank you.